anyway, Jorge's going to give us a brief update on what they've been working on at Harmony Biosciences, which is a new group in our organization, and you can use either the handheld there or that one.
Excellent.
And we can't wait to hear this. I don't even know what he's going to say. No, I'm just kidding. I do.
Excellent. Thank you. Well, I heard today a lot about the trials and the foundation, LGS Foundation, and I've interacted with a lot of you, and we are honored and privileged to be here today to make an introduction, basically, for potentially a new player in the field, with a new opportunity in treating developmental epileptic encephalopathies like Dravet syndrome and LGS. So first, the introduction of the company. Harmony Biosciences is a small, relatively small, young, but very robust, I would say, biotech. Was founded 7 years ago, and it's focusing on developing drugs for rare neurological, mainly pediatric diseases. Our latest effort is, in fact, in rare refractory epilepsies like Dravet and LGS. And I will go through the pipeline. So again, young company, but with a lot of potential. We have already an approved medication for the treatment of hypersomnia associated with narcolepsy. And we have a big franchise on sleep-wake symptoms, diseases like narcolepsy that I mentioned, but also hypersomnolence associated with other neurological diseases like myotonic dystrophy and Prader-Willi. We expand from sleep and wake diseases to neurobehavioral diseases like Fragile X, and we use as a drug a synthetic cannabidiol, which to you here is perhaps not that unknown with the development of Epidiolex. This is a different synthetic cannabidiol that we are developing currently for the treatment of Fragile X. And then we're focusing, of course, with a new compound that has a history, and I will go through it very quickly, on, as I mentioned, rare epilepsies like Dravet and LGS. So first, a little bit about the compound EPX-100. This is a compound that we acquired, Harmony Biosciences, recently, a couple of months ago, 3 months ago, acquired another small biotech, Epigenics, that has been developing a drug, a repurposed drug, a drug that was used many years ago as the first generation antihistamine that was basically discontinued, not because of any safety concern, but because it was the first generation and there were new antihistamines that were developed that were better with a more favorable profile, dosing profile. So this compound, clemizole, has been studied extensively by a group of scientists in University of California, San Francisco, not Santa Barbara where I live, but San Francisco, in Scott Baraban's lab. And he has an animal model, and I think it's important to understand why I spent a whole slide on this. So using an animal model, a zebra fish genetic model that has high throughput. He can test several compounds in a very short time. This model is based on genetic manipulations, but also heavily on electrophysiological findings and behavioral even findings that resemble seizures in animals and in humans, which is remarkable. So he manages to breed the activities that are seen in an aquarium, let's say, to the bench. And that is very, very important. What he has done, he has tested several compounds that are active in controlling seizures in humans, and he has seen activity in one model with one genetic manipulation model, the SC1 mutation. This is the same mutation that is found in most patients with Dravet syndrome. So he tested a lot of compounds that are available, and those that work in that animal model also, that they work in the clinic, they also work in that animal model. While similar mutations in rodent mice, you see that not all compounds that work in the clinic work in the mouse model, but they do work in this zebrafish model. This is what we call in the science business, let's say, a high predictive validity of an animal model. Again, this is very, very important. So based on this model, he found out that this compound, clemizole, has a very good anti-seizure activities, also affects behavior induced by seizures in this zebrafish model, and even EEG signs. So based on this model, what he did, he tried to understand how this compound, clemizole, affects its anti-seizure activity. And he found that clemizole works in this model not because it is an antihistamine, but rather because it affects serotonin receptor, 5-HT2B, 5-HT2C, and especially 5-HT2C receptors. And we will hear by Dr. Ray following my presentation a little bit more about 5-HT2C receptors. So this compound is a direct agonist for this serotonin receptor. By the way, these are the serotonin receptors that are also affected indirectly, primarily, or directly as well by Fintempla, fenfluramine. So there is a good scientific basis to be very, very excited about clemizole in treating seizures associated with Dravet and also LGS. This is a typical, I would say, clinical trial design. Patients that are already on anti-epileptic drugs, but they do still experience seizures, will be screened. We will establish a very good baseline, and then baseline for seizure activity. And then they will be randomized to receive either placebo or the drug EPX-100, clemizole that is, for 12 weeks. And then there will be an open label extension, as we call it, where for almost 3 years, or a little bit more than 3 years actually, people will receive either, we will only receive the medication, EPX-100. So this next slide, and this is, I think, I do the next one here. This is what I have to share with you. Again, a brief introduction to the company, a brief introduction to clemizole, and we hope in a way that we will be able to offer in a few years a new opportunity for treating refractory seizures associated with LGS. Thank you for your attention, and thank you very much for allowing us to do this presentation. Thank you.